Hello, my name is Jordan Hess. I hold a master's degree in neuroscience from the University of Texas at Dallas. Today, what we're going to be looking at is some research that's coming out of Johns Hopkins from researcher Dr. Yuto Asagawa from the Camille Lab. These researchers are looking at whether adolescent cannabis exposure may lead to lasting social deficits. And they're going to be looking at the medial prefrontal cortex, and they're going to be looking for depletion of the principal immune cell of the brain, microglia. The type of genetic variation that the researchers were looking at falls underneath a broader category known as copy number variations that can entail duplication or deletion events. So we need to review some chromosome anatomy. And so if we divide the chromosome into left and right halves, each one of those will be referred to as the sister chromatids, bound in the middle by the centromere. Above, we have the short arms called the P arms for petite or small, and below we have the Q arms for Q, meaning tail. So here we have a 16p11 duplication in these animals, and so that's on the 16th chromosome in the P arm at the 11th position. Okay, and this is a duplication event. So these copy number variations can entail deletion events or duplication events. And again, we're looking here at approximately 27 to 29 genes that are being affected. And so there's some overlap between these conditions. As you can see, both confer a risk for autism spectrum disorder, intellectual disability, epilepsy, seizures, congenital abnormalities. And so there are some conditions that are limited specifically to duplication events, and this is something of interest to us here for schizophrenia and bipolar disorder. And so two things kind of come to mind, right? When I see this, I think to myself, well, there could be additional genetic factors that, like in other words, what's the reason that not everybody is going to get it, right? But there's an additional factor, right, of lifestyle choices. And so that's where the researchers are really taking a look. And that's going to be the method that's largely explored in this paper. Okay, before we begin, we need to get into some microglial biology. And so microglia have a ramified or resting state, which is indicated by extensive branching, and they have an activated state that is associated with an amoeboid-like morphology and also a spheroid-like morphology. So this is hippocampal slice tissue over the course of 12 hours, and so the simple act of slicing will be sufficient to cause microglial activation. This particular stain here also stains for blood vessels. So the blood vessels are going to be indicated by the arrowheads, where the microglia will be indicated by the arrows. And so we can see this again more clearly here. Over the course of 24 hours, the microglia progressing from a resting or ramified state to a more activated state with the spheroid and amoeboid-like morphologies. All right, and so in terms of microglia activation, the question comes to mind of how and why are these microglia being activated? So when we have tissue damage, nucleotides are going to be released, specifically ADP, which is going to bind to the surface of the microglia to purinergic receptors to the P2Y12 receptor, which is encoded by the P2RY12 gene. And so red fluorescence is going to indicate the presence of the P2Y12 receptor, whereas green fluorescent protein will indicate the presence of microglia as it is expressed under a microglial specific promoter, specifically CX3-CR1. Glial fibrillary acidic protein will indicate the presence of astrocytes. Since we're looking at light, green light plus red light equals yellow light. So here, where you see yellow, this indicates microglia that are expressing the P2Y12 receptor. All right, so these are cultured microglia in a gradient of ATP. And previously, I had mentioned that when you have an injury, nucleotides like ADP are going to be released, and then you'll have a microglial migration towards the site of the injury. And so, well, activation and then migration. And so, over the course of 5 minutes, that's kind of what you're observing here when ATP is being applied in vitro. So you'll see a little bit of membrane ruffling occurring, right? So that's indicating some activation of the microglia as they progress towards this polarized state where they will be moving towards the highest concentration of ATP, which would of course, be the injury if this was in vivo. And in the case that this gene is knocked down, there's no membrane ruffling and there is no polarization. And some interesting notes here. So right here, if we look at this first image and we look at the middle panel over the course of 0 to 24 hours, as the microglia become more activated, the expression level of the P2Y12 receptor decreases. And this tells us two things, that, that it's only the P2Y12 receptor is only like necessary initially, and that it can be used as a marker for microglial activation. And that's an important point because we'll be able to use that when we're going over the research from the Hasegawa paper. And so here, the researchers are looking in vivo with a technique known as focal laser ablation. So they're going to destroy some tissue, and normally there's going to be microglial process extension towards the area of tissue that was ablated. But in the knockdown animals, this process extension towards the injury is limited. But if we look over the course of 120 minutes, we see some process extension, and this tells us that in the absence of the P2Y12 receptor, that there are compensatory mechanisms in place for the microglial activation. So the researchers are administering THC subcutaneously, and they want to look at the expression levels in different cell types. And so here on the left, the researchers are performing flow cytometry or fluorescence activated cell sorting, in which they're using some specific markers to identify the different cell types. And so Nguyen for neurons, CD11B and CD45 for microglia, along with TMEM119 and ACSA2 for the astrocytes. And they find that the expression of the CNR1 is as follows: highest for neurons, second highest for astrocytes, and lowest for the microglia. And that's the population that is of interest in this paper. So the researchers here are wanting to validate their animal models, right? And so this is supposed to be so this is the LOXP system, right? So the researchers can knock down a gene. They're going to be wanting to knock down the CNR1 gene, but they're going to want to knock it down selectively in the microglia. That's that microglia specific promoter that we talked about, the CX3CR1. Now, the Cre-LOXP system, right? So that's a recombinase that allows some DNA to be removed. And so instead of it being ubiquitously expressed in the animal, it's expressed in the microglia. And in this case, the researchers are also going to have temporal control of it because they are going to create a chimera protein with mutated estrogen receptor, where the Cre recombinase will become activated upon administration of the estrogen receptor agonist tamoxifen. And when they do, they see that there's selective knockdown of this gene in microglia, but not in astrocytes or in neurons. And when the researchers look in terms of protein expression, they find that this is also true. So IBA1 is our microglial marker along with CD11B, and there is this expression of CNR1 which is knocked down in the floxed animals. So in terms of what the researchers are looking at here, so they're at postnatal day 30 to postnatal day 51, so a period of 21 days, the animals are administered THC, and there's some microglial phenotyping that is occurring. Here they're looking in the medial prefrontal cortex, so you have IBA1, which is your microglial marker, and then you have this gene, which is encoding for the P2Y12 receptor. And in its presence is going to indicate the degree to which the microglia are activated. So when we look at the THC wild type and the 16p11 duplication vehicle conditions, what we're going to notice is that these two conditions are going to tend to parallel each other, in that whatever effect we see in these conditions will be exacerbated in the 16p11 duplication THC gene environment interaction condition. And so here we're seeing some depletion of microglia, so there's a decreased abundance, but we're also seeing some increased activation in addition to a further exacerbated decreased abundance in the gene environment interaction condition. All right, so in the previous slide, researchers were looking at many cells, and here we're looking at single microglia cells. And what we notice when we're looking here is we see that there's a decreased extensiveness of the branching and an increased size of the cell body relative to the size of the entire cell, and that this is worsened in the case of the gene environment interaction condition. So do those changes in microglial morphology correlate with an increased microglial activation? The researchers looked in the regions of the medial prefrontal cortex, ventral and dorsal hippocampus, nucleus accumbens, and amygdala. And only in the case of the medial prefrontal cortex did the research in the gene environment interaction condition did the researchers find increased microglial activation. Okay, so here we have the P2RY12 positive cells indicating the resting microglia, and the P2RY12 negative cells indicating the activated microglia. And so when we see a decrease here in the number of resting microglia, we would expect to see some corresponding increase in the number of activated microglia. But when we see a decrease in both the resting microglia and in the activated microglia, this is indicating some decrease in microglial abundance. For the decreased microglial abundance, researchers wanted to determine if this is being indicated by apoptosis, which is a controlled form of cell death that is not marked by inflammation, or necrosis, which is an uncontrolled form of cell death that is marked by inflammation. And so here in this lower panel, the green is indicating the apoptosis marker apoxin, where red would indicate the necrosis marker 7-AAD if present. And so these are cultured microglia. The microglia were isolated with the technique of magnetic activated cell sorting as opposed to a fluorescence activated cell sorting. And this technique was more efficient in that of the CD11B positive cells, approximately 95% of those cells that were isolated were microglia. And what we see here is that THC is a sufficient driver of apoptosis, and that this apoptosis is further exacerbated in the case of the gene environment interaction condition, but this is not true for necrosis.
All right, and the researchers wanted to determine whether CNR1, cannabinoid receptor type 1, or cannabinoid receptor type 2, CNR2, is the primary driver of microglial apoptosis in the medial prefrontal cortex. So again, this is some cultured microglia from knockout animals where the CNR1 or the CNR2 is knocked out. And when the CNR1 here is knocked out, the apoptosis is eliminated in the case of THC being administered. But this is not the case when CNR2 is knocked out, indicating that CNR1 is mediating the microglial apoptosis. Okay, and so this, what the researchers had looked at in vitro, they wanted to verify in vivo. And so in terms of apoptosis, we have what's referred to as a point of no return. And so caspase 3p17 serves as a marker before the point of no return, so cells that are leaning towards apoptosis. And DNA fragmentation tunnel serves as a marker for after the point of no return, so cells that are committed to apoptosis. And what we're seeing here is that there is some increased abundance of microglia that are leaning towards apoptosis and that are committed to apoptosis in these conditions, and that is being worsened in the case of the gene-environment interaction condition. And we see a quantification of that here. And again, just want to draw attention to the fact in the 16p11 duplication itself is a sufficient driver of microglial apoptosis in the medial prefrontal cortex. Very important point there. And so in terms of what's happening inside of the cell to drive the microglial apoptosis is transcriptome profiling. So it's known as an ingenuity pathway analysis, where this is a functional gene interaction network that is used to identify upstream regulators or potential upstream regulators. And so here they were able to identify TP53, or rather the activation of TP53 through the inhibition of NOP53 to be the predicted primary driver of microglial apoptosis. Okay. The researchers looked at TRP53 expression levels in the various experimental conditions and found a parallel between the apoptosis markers from the in vivo experiments. When the p53 inhibitor was applied, the apoptosis that would otherwise be occurring was eliminated, with the nuance, the minor nuance here in the case of the 16p11 duplication condition. Okay, and here the researchers are interested in learning if some abstinence period of, so the 21 days. So the researchers administered THC for 21 days. Now they're looking at a subsequent 21-day abstinence period, and they're wanting to ascertain whether there are any lasting effects on behavior. All right, so here we have what's referred to as the three-chamber social interaction test. So think 10, 10, 10. Okay, so an animal will be placed in the middle chamber and allowed to freely explore all three chambers for a period of 10 minutes. Then at which time the animal will be gently guided back to the center chamber, and a stranger animal of the same species will be placed into the plastic cage, and the animal will be allowed to again freely explore while time spent sniffing the stranger will be recorded. Afterwards, the animal will be gently guided back to the center chamber while an inanimate object will be placed into the other plastic cage, and there will be a final 10-minute period of exploration, and the time spent sniffing will be recorded. As is indicated by these heat maps here, typically it will be the case that the animals will have more interest in the live animal than in the inanimate object, and that this is also true in the gene-environment interaction condition, so really no effect being observed here.
Now, if instead of placing an inanimate object, so it's the same procedure, but instead of placing the inanimate object, a second stranger of the same species will be placed. Normally, animals will spend more time with the new animal. But in the case of the gene-environment interaction condition, this is not the case. In fact, even in the 16p11 duplication vehicle condition, we're seeing a decreased interest in the new animal. And that's how we're getting these negative values here when we look at the discrimination index. So when the time spent sniffing stranger 2 decreases, this expression will become negative. Okay, and so here we have what is referred to as the 5-trial social memory test. And so a subject will be introduced to a litter mate, and 10 minutes later will be re-exposed and so forth on the order of 10 minutes for the second, third, and fourth trial. And typically what will occur is a habituation. So they get used to the animal that they're being introduced to. And then on the 5th trial, a new animal's being introduced, and we'll see a spike in interest. And when we look in the case of the gene-environment interaction, what is happening is that there is some the habituation that's normally observed is not being observed, and in fact, instead of like an increased interest when the new animals being introduced, it almost seems like a decreased interest. And so some important point to consider here is that, you know, one might say, well, maybe this is due to a decrease in locomotion, and so the researchers looked at the open field test and determined that that wasn't the case, and they said, well, maybe this is due to increased anxiety. And so the researchers looked at the extended plus mazes and found that this was not the case either. So for here, the researchers are looking at 2 additional tests, the novel object recognition test and novel place recognition test. And so for these, the researchers are going to place the animal into an open field arena where the animal will be allowed to habituate for 10 minutes. Okay, each day for 3 days. Okay, after the final habituation period, an hour will pass. 2 objects of the same size, shape, and color will be secured to the floor, and you'll have your initial training period where the researchers will record the total amount of time spent sniffing each object. There'll be a 15-minute intermission, at which time one of the objects will be replaced with an object of the same size but of a different shape and color. And the 5-minute retention period will take place where the time spent exploring the new object will be recorded. And so in the case of the novel place recognition test, one of the 2 familiar objects will be moved to a novel location, and the time spent in that novel location will be recorded. And in both of these tests here, no effects were observed. So one might wonder, okay, so is there some physiological correlates, or what are the electrophysiological correlates in the medial prefrontal cortex to the behavior that's being observed? Because, and we'll kind of look at this at some point later in the presentation, but the microglial population during this abstinence period is going to rebound. And so the effects that are being observed are likely due to the fact that microglia are shaping these synaptic connections through pruning and remodeling during adolescence. And so when the researchers look in layer 5 of the medial prefrontal cortex and they are administering a hyperpolarizing current, the voltage traces that are recorded will indicate whether we have pyramidal tract neurons or intratelencephalic neurons. Here the researchers are looking at pyramidal tract neurons, and this is in response to an injected depolarizing current. And here we have some action potentials, and what can be seen in the case of the gene-environment interaction condition is that there is an increased current that's required to elicit the action potentials. And so here we have some different metrics. So one of them is the input resistance, and that's how easily that the membrane voltage is going to change in response to an injected current, or this slope here that is occurring before the action potential. And so a decrease in the input resistance is related to a decrease in the excitability of the cell, where for the rheobase is defined as the minimum amount of current that is required to elicit an action potential when applied for up to an infinite duration. And so when more of the current is required to give that action potential, then we have an increase in the rheobase. And so that's going to make it harder for the action potentials to occur, and we're going to have a corresponding decrease in how often that the action potentials are occurring, or a decrease in the spike frequency. So what this translates to is that overall decrease in the excitability of these neurons. When the researchers looked at intratelencephalic neurons, they found that these neurons were not affected in the experimental conditions. And so let's just return for a moment to the idea that, previously all the animals were not administered THC, right? So we had the wild type condition and we had the 16p11 duplication condition where the animals were not administered THC. Here, all animals will be administered THC. And here, they're knocking down the CNR1 gene in these floxed animals in the gray and blue bars. So here, what we have, this is from a previous image, and it's indicating the true control condition in which you have a wild type animal with no THC being administered. And this is in the medial prefrontal cortex and indicating a rich microglial abundance. In the case of THC being administered, we see the decreased microglial abundance along with the corresponding changes in morphology in terms of decreased branching and the increased size of the cell body relative to the entire cell. And we see that the the decrease in microglial abundance is reversed in the case that the CNR1 gene is knocked down, and the same follows for the changes in microglial morphology. Okay, and here one might have the kind of question that would come to mind is, when the CNR1 gene's being knocked down, why is there not a return to the control values, right? Well, there is, right? But this control is with THC being administered, but in the true control is the wild type with no THC being administered, and that's what these bars are actually pushing towards. And we're going to see that trend as we look through these papers. So what we're seeing here is increased microglial abundance in these first two images. And then in this last image here, what we're seeing is that this increase in microglial activation that is observed in the case of the gene-environment interaction condition is ameliorated in the case that the CNR1 gene is knocked down. Okay, so in terms of the in vivo measuring of apoptosis, so if we look at so the first panels and then the third panels, those are the cases in which THC is being administered, and we're seeing more cells that are both leaning towards apoptosis in the upper panels and in the lower panels that are committed to apoptosis. And in the case that of the CNR1 gene being knocked down, the microglial apoptosis is being eliminated. And here's a quantification of that. So see that the gray and blue bars have been eliminated here, indicating the reduction in apoptosis. Okay, and so the apoptosis is being
governed by the CNR1, but what about the electrophysiological effects that were observed on the pyramidal tract neurons, and what about the effects on behavior? And so here, what is observed is that decreased excitability that was observed in the case of the gene-environment interaction condition is reversed. So, right, so this increased input resistance indicates an increase in excitability, and the same is true for the decreased rheobase here and the corresponding increase in spike frequency. When the case of social novelty is examined, let us return for a second to our previous results, where it was determined that the 16p11 duplication vehicle condition animals exhibited some effect here, and that in the case of the gene-environment interaction condition, there was a much more pronounced effect. And here, when the CNR1 gene is being knocked down, there is that similar return to control conditions, but one might be quick to say, well, why isn't this blue bar, why isn't this condition returning to control levels as was previously observed in the last few slides? And well, the reason for that is it is returning to a control, but just a different control, right? Because the control here is the 16p11 duplication vehicle condition, and that is where it is returning to. Here for the 5-trial social memory test, let us recall that there was some habituation over the first 4 trials, and then when the novel animal was introduced, there was an increased interest, but in the case of the gene-environment interaction condition, over the first 4 trials, there was no habituation, and then that apparent decreased interest upon introduction of the novel animal. So here the line of importance is this blue line, and so the blue line is indicating that there is indeed a return to control conditions in the case that the CNR1 gene is knocked down. All right, so as was previously mentioned, this region for this copy number variation, it contains approximately 27 to 29 genes, and the researchers want to take a look at these genes to determine the genes that are playing a primary role. Okay, as I pointed out before, schizophrenia and bipolar disorder are being indicated by the duplication event, but not by the deletion event. And so if the researchers were to next study deletions in the context of this paper, and they looked at the microglia, they looked at microglial abundance in the medial prefrontal cortex, and they looked at changes in morphology. Let's say that they saw no difference, okay, in the case of deletion events. That would indicate that the changes that are being observed in this paper are more strongly linked potentially to schizophrenia and bipolar disorder, whereas if they were to, say, observe very similar changes to what they observed with the duplication event, this would potentially indicate some overlap between conditions like autism spectrum disorder and intellectual disability with schizophrenia and bipolar disorder. All right, so let us recall that in the case of pyramidal tract neurons, there was a decrease in excitability that was the most pronounced in the case of the gene-environment interaction condition, and that in the case of intratelencephalic neurons, these changes were not observed. So the first question that the researchers have is, why are the pyramidal tract neurons being affected, but the intratelencephalic neurons are not? The second question that the researchers have is, how are these changes in the excitability of the pyramidal tract neurons in layer 5 of the medial prefrontal cortex having or exerting their effects on social memory. So an additional future direction that I have is, so I'm looking at the abstinence period, I'm looking at the microglia that are rebounding, and I'm thinking to myself, right, that what if the mechanisms for microglial rebounding were impaired? How much more significant would the social deficits that were observed be? That's one question. And then the additional question that comes to mind is there's many hypotheses for diseases like schizophrenia. And so one of those hypotheses is, for example, that something like mono in some individuals might trigger the onset of this disease. And so one might kind of, after reading this paper, be curious if the mono was able to affect, say, the medial prefrontal cortex, and the principal immune cell microglia was depleted. Could it be that this virus now could have a much more significant and detrimental effect? Okay, so looking at the synopsis here, so we have some male mice with the 16p11 duplication that are going to be administered THC during adolescence. The THC is going to bind to CNR1 expressed on the surface of the microglia in the medial prefrontal cortex and initiate apoptosis via TP53. In terms of what's observed in adulthood following an abstinence period, right, is there's social memory deficits and there's decreased excitability of the medial prefrontal cortex, layer 5 pyramidal tract neurons. And so kind of in terms of a bigger, broader picture, it may come to mind that for a very small amount of individuals that are consuming cannabis, that this may trigger in them some social deficits up to and including schizophrenia and bipolar disorder. And so hopefully this paper is shedding some light on some potential mechanisms by which that may be happening. And so an important note here is that, say, in conditions like schizophrenia, you have in females having some increased protection or resilience towards this condition. And so here the researchers found that with this gene environment interaction between 16p11 duplication and THC, that the females were relatively unaffected, right? So this is a fact that's isolated to male mice. Okay, so that's something important to note. I'm definitely interested in seeing future developments from this lab, and we'll be following this closely. Thank you. Until next time. All right, don't forget to like and follow if you would like to see more content. Thank you very much.